How is everybody doing? Good to see at least the people who are already here survived Snowpocalypse 2026. Right, right. I had a little bit of snow at my house, but yeah, it was mostly just freezing rain for sure. Um, and then of course, what was on the roads was pretty much just a solid sheet of ice. All right, so as you can imagine, all of my classes' schedules are all over the place, and so please, if you will, remind me what we stopped with last time. Acids and bases, and we were going to go ahead and continue. Okay. Okay. So that's good. I can get that out of the way, and we can go ahead and pull some of this stuff up. No, why I didn't get the Monday, Wednesday schedules on here that I can derive from this. Oof. Okay. And for lab, we had gotten about halfway through connective tissues and blood. Yeah. Awesome. Thank you for the refresher. We'll get my head on straight. All right. Well, unfortunately, that means Monday we should have finished that biochemistry, and today we should be getting into cell structure and transport. And so where we were a little bit ahead, we are now essentially a full lecture behind schedule. Yeah. So, I draw on this. That is the most terrible arch I've ever seen. Oh, that's not going to work either. Well, maybe that's about as clear as mud. Okay. Now I am trying to record these lectures using Teams. So if you will please bear with me for just a second while I try to figure that out. Okay. That looks like it'll work. All right. Want to go up top? Would you mind hitting the lights for me, please? At least the lights up here in front. If we need to leave something in the back on, that's fine. And I'm not sure which switch is which, so good luck. I appreciate it. Well, last time it was one of those switches on the wall right there, turned them off. Okay. It's like slam them all. No? Okay. I don't know. Something must have gotten changed. That these are three-way switches when they're not wired. Do that from the other side. All right, so real briefly, I wanted to go back. I don't feel like I did a really good job explaining redox reactions to y'all. So I'm going to go back and hit that again, right? The oil rig thing still applies, right? Oxidation is loss of electrons, and reduction is gain of electrons. But we can think about it oxidation by losing hydrogen or gaining oxygen, right? And that sort of makes sense. To be oxidized, to add oxygen there. So if we take a look at the glucose, the C6H12O6, it is oxidized, right? The hydrogen atoms in there are pulled away and turned into water, right? Now, it's important to remember that this is a summary of many different chemical reactions. So like all of glycolysis, all of the citric acid cycle, so on and so forth, is contained within that little arrow, right, to the right in this chemical reaction. And then we have 12 oxygen atoms over here on the left side, right? And 6 of them get pulled over to join the leftovers of glucose. So 6 CO2, 6 times 2 is 12, right? And 6 of them remain and gain that hydrogen that got pulled off of the glucose. Okay, so in both instances, right, if you're familiar with the electron transport chain in the mitochondria, and that's something you've studied before, right, essentially what we're doing in glycolysis and the Krebs cycle is we break glucose apart into a bunch of little pieces and we create a bunch of other small molecules. And then we run them through that electron transport chain to pull the electrons off of them, oxidize them in that way to create more ATP, right? And again, all of that is sort of subsumed within the little arrow here. And we're not going to get into that too much. So as we see this hydrogen being pulled out of this and being transferred and used to form water, right? That's how we're going to call this oxidized. So the important thing to remember is that we're breaking down the substrate here and transferring energy to other compounds. All right. I hope if there was a little bit of confusion, excuse me, about that, that helped clear it up. And as always, if you have any questions about any of that, then send me a message on Blackboard or ask me after class. So acids and bases, right? Electrolytes, meaning they dissolve in water and exist as ionic compounds, meaning they are going to have a positive or negative charge, right? So acids, right? They donate hydrogen ions into the solution that they're dissolved into, right? We write that as H+. That is hydrogen missing its electron. And as you might imagine, that's going to make the hydrogen very ready to interact with other chemicals, other molecules. It's going to be a very willing electron recipient, right? But we flip that on its head. We're saying we're donating protons in this case. So acids donate protons in chemical reactions, and they're going to bind negatively charged ions. And bases are just the opposite, right? They will bind proton, they will accept protons and remove protons from a solution here. So sodium hydroxide, right, is an example of that. It will dissociate in water to sodium and a hydroxyl ion there, and the OH negative is very happy to accept that extra proton and become water and become electrically balanced here. Okay. Now, you've probably heard of the pH scale. It means potential of hydrogen. And it's a concentration of hydrogen ions or protons in solution. It's a logarithmic scale, right? And so each whole number, each integer that we increase or decrease is a multiple of 10 times, 10 times. So as the concentration of protons increases, acidity increases, and then so we're going to move downward on the pH scale, right? And as the concentration of protons decreases, the alkalinity increases or acidity decreases, and the pH is going to move upward. So on, oh, let me see. There we go. All right. So this slide gives us the pH scale, right? Further to the right, right? And you can see that corresponds as we move to the left to the scale written with the little brackets around our proton, right? Around our H+ ion. And then to the left of that, we have concentration of hydroxyl ions, the OH negatives, right? That would be the scale POH. It is a scale that exists. We don't use it very often, right? But that does exist. So for our purposes, we're concerned with the pH scale. The POH scale works the same way, just inverted, because it's measuring the potential of those OH negative ions rather than our protons there. So in the pH scale, a pH of 7, right, would represent something that is neither acidic nor basic. It is perfectly neutral. So like a pure distilled water that doesn't have anything dissolved in it would be a perfect pH of 7. And then the acids are going to be progressively further down the scale. So something like milk is slightly acidic, right, with a pH between 6.3 and 6.6, right? The hydrochloric acid in our stomachs, right? We concentrate to a pH level of 2, right? Or if we had pure 1 molar hydrochloric acid, that would have a pH of 0, right? So how do we compare them? Again, each whole number is a 10 times difference. So if we move from lemon juice at a pH of 2 to 1 molar hydrochloric acid at a pH of 0, right? 2 to 1 is 10 times, 1 to 0 is 10 times, 10 to the second power is a 100 times difference in acidity. Make sense? Yeah? Okay. Just want to drill that in, right? It's not 10 plus 10, it's 10 times 10, right? And we can cross 7 with that, right? So if you compare egg whites at a pH of 8 with black coffee at a pH of 5, how much more alkaline is the egg white? So from 5 to 6 is 10, 6 to 7, 100, 7 to 8, 1,000. So that's a 1,000 times difference, right? And we can compare alkalinity or acidity depending on where it is in the scale and which direction we're moving. I don't see any smoke coming out of anybody's ears yet. Am I good to move on? Okay. Let me do that then. So when we mix acids and bases, they react with each other. As you might imagine, right? One has a high concentration of free protons and the other has a high concentration of free OH negative groups, right? And those positive and negative charges are going to interact. We're going to form chemical bonds and they're going to neutralize, right? They're going to balance each other out. You've probably seen this reaction happening in real life. If you've ever seen a movie or done the stereotypical science fair volcano project, and you've mixed your baking soda with vinegar, right? That is an acid-base reaction. It neutralizes and forms some carbon dioxide gas and water and some salt. The example we have on the screen here is the hydrochloric acid in our stomach with sodium hydroxide, which I believe we would call lye under normal circumstances, right? Those are going to react and they're going to form table salt and water, right? Now, why do we care about pH in the human body? Well, because the enzymes inside of our body are proteins, and increasing or decreasing the pH can cause these proteins to change shape. And if a protein changes shape, because form and function are inextricably linked, the protein is not going to perform its job as efficiently or at all any longer. And then if it's an enzyme, that means it's a catalyst that's helping to speed up chemical reactions, sometimes billions of times, right? Remember, some reactions that would normally take 10,000 years to happen once happen several thousand times per second in the body because of these catalysts. And that means we stop living, right? You'll find that that's the endpoint for a lot of things that might go wrong, right? Incompatible with life, as we say here. So even slight changes in pH can permanently damage cell components or cause the enzymes we need to keep using oxygen to keep ourselves alive to stop working. So generally speaking, our body is slightly alkaline, right? Particularly in the bloodstream, right? The blood in the veins is slightly more acidic than the blood in the arteries because there's more carbon dioxide dissolved in the veins than there is in arterial blood. And if you'll recall, carbon dioxide dissolved forms carbonic acid. Now, we have some buffer systems in the body that will help maintain a pH level despite the addition of acidic or alkaline substances. So it helps to buffer or stabilize the pH level, both in the intracellular fluid, right, that's inside the cells, or the extracellular fluids, including the blood, right? We have a limited amount of alkaline substances in the body, right? We call that the alkaline reserve. So we can add acidic compounds and buffer it and stabilize it to a point, but then once that is used up, we have no more alkaline reserve. We'll start to see rapid pH changes in the body unless it's accommodated by other mechanisms, right? We have an almost unlimited supply of acidic compounds in the body, and it's produced primarily as an end product of metabolism. And so if we have some circumstance where we have a large amount of alkalinity added to our body, generally speaking, so long as it's not too rapidly, our body is more capable of accommodating that because we are continuously generating acids that would neutralize those alkaline substances here, right? So there are some examples of some acidic substances down there at the bottom of this slide here. Now, the three ways that we deal with primarily acidic substances. Most of these systems are concerned with buffering acids primarily, although they do work in both ways. We have chemical buffer systems that exist within our body fluids and blood, and those are available for instantaneous chemical reaction. The brainstem has chemoreceptors in the medulla oblongata that monitor the pH level of blood and can change how fast we're breathing and how deeply we breathe, right? By blowing off more carbon dioxide, by breathing more rapidly, we are taking acid directly out of the blood, removing it from the body entirely, right? And then finally, our kidneys, right? By changing the concentration of waste products in the urine and changing how much we reabsorb from the urine, right? We can dump a lot of metabolic wastes and acids out, right? Very powerfully. But these are also rated in terms of speed, right? The respiratory mechanisms take several minutes to change. Renal mechanisms can take hours or days to make large changes, although they are the most powerful system. So first, we're going to talk about chemical buffer systems. Again, these are the fastest rapid response because these represent agents within the body fluids that are there, able to react instantaneously with anything added. So we need something that's going to resist large swings in pH. Now, we have strong and weak acids. A strong acid is not really a good candidate for a buffer because a strong acid dissociates completely in water as soon as it's added, and all of those free protons are then donated to the solution, causing a large pH change, right? But a weak acid, right, as demonstrated over here on the right-hand side, part of our bicarbonate buffer system here, is only going to dissociate partially. And by dissociating partially, it's going to donate a few protons, but also leave those regions with negative charges exposed. So if more acid is added, right, those negative charges are available to take protons out of solution, and in that way, they buffer the pH and prevent it from falling as quickly as much as it might. Okay, so when the pH drops, that represents an increase in protons in the solution, right? The chemical buffer is going to bind more protons, right, in order to pull them out of solution. When the pH rises, that's going to represent a decrease in the number of protons in solution. The chemical buffer will release. More of it will dissociate, releasing more protons in order to keep the pH from rising too quickly. The most important one is the bicarbonate buffer system. That's what we're going to spend our time talking about today, right? So the bicarbonate buffer is a mixture, right, of carbonic acid, H2CO3, and a weak base. So salts of bicarbonate, right? And you can see the difference in the chemical formula between carbonic acid and that bicarbonate there is merely the removal of a single hydrogen atom here. This works in both intracellular fluid inside the cells and extracellular fluid, so all of the water in the tissues as well as in the blood plasma, right? This is the only important one for extracellular fluid, and it's very important in the blood. So this little diagram right here, this little chemical reaction, notice we have arrows pointing to the right and we have an arrow pointing to the left as well. Right, that denotes that this reaction is reversible, which is exactly what we want in a buffer system. Right, we want it to be balanced such that a small change in the chemical's environment is going to cause it to move back and forth, right, and react one way or the other. On the left side, we have our carbonic acid, our H2CO3. And on the right side, right, we have broken that apart. We've pulled a proton off of it. And now we have our bicarbonate plus a proton. So this buffer system exists in the bloodstream. As we add acid, this bicarbonate buffer over on the right-hand side that is dissociated, right, is going to accept the additional protons that are added to the system, and the reaction is going to proceed to the left, forming our carbonic acid, right? So now in real life, what do? We breathe out, right? The carbonic acid, the CO2 dissolves out of the bloodstream, right? It is removed from the bloodstream. We release it as CO2, restoring, right, removing that acid essentially from the blood, and then the buffer can move back to the right, right, and become a proton acceptor again, right? And this remains true, right? We can add hydrochloric acid directly to this fluid, and we'll only see a very small change in pH until all of that available bicarbonate on the right-hand side is used up, right? Once we convert everything on the right-hand side of that equation to carbonic acid, there's no chemical buffer left. And so any additional acid added to the system is going to cause the pH to drop rapidly, right? There's no more buffer left, right? That's our alkaline reserve that I mentioned earlier. There's a limited amount of that. Now we can regenerate it, but it's slow, right? We get some of that through our diet, so on and so forth. Now, if, on the other hand, we add sodium hydroxide, lye, something very alkaline to the system, right? That weak acid over on the left-hand side is going to proceed to the right, right? The arrow points to the right, and that hydrogen ion, that proton, is going to separate out, right? And it's going to buffer that alkalinity that we added, right? So same deal. We can move back and forth, back and forth. Oh, too far, too far. So that's the bicarbonate buffer system. Now, proteins. Now, have we looked at amino acids already in a previous lecture? No? Okay. No? Okay, cool. We'll get there. All right, so proteins are made up of subunits called amino acids, and amino acids have an amine group, NH2, I believe, and then a COOH group, right? That's the acid. And that is to say, different regions of amino acids are going to have exposed positive charges or exposed negative charges. You can think about it that way. And so if a protein is made up of 1,000 amino acids among 20 some odd different ones combined in different orders, the proteins are going to have regions that will accept protons, right, and act as a weak base, and they'll have regions that will donate protons or act as a weak acid. And that way, the term for this, a molecule that can function as both a weak acid and a weak base in solution, is to say it's amphoteric, right? And so plasma proteins, intracellular proteins, both carry this quality, and so they can assist in chemical buffering here, right? So in much the same way that the carbonic acid works, right? The COOH group in the amino acids is going to release a proton when it needs to, and when we become too acidic, right? Those amine groups will bind an additional hydrogen or additional proton there. So there we go. All right. So the buffer systems. If we're buffering acids or buffering alkaline substances, bases would be the other word for that, those compounds still exist within the system, right? They're still there. We're binding them up so that their effect on the pH of the fluid doesn't change dramatically, but they're still in the body, right? As we move into the respiratory or the physiological buffering systems, right? These are going to work with the chemical buffers, but then at some point, we're actually going to remove the substance from the body, right? So, as it says here, our chemical buffers are not going to eliminate them from the body. They're just going to bind them and hold them until either our lungs or our kidneys can remove them, right? So again, as we breathe out, volatile carbon dioxide is expelled, right? That's essentially taking that carbonic acid out, right? And leaving water behind. And then we already mentioned how kidneys are going to dump a lot of these acidic compounds from our body here, right? And so this chemical formula, right? This is a set of 3 different formulas. The right-hand side of it represents that bicarbonate buffer system that we've already looked at here, right? And then the left-hand side represents physiological processes, right? So on the left-hand side, that carbon dioxide, that carbonic acid, could be contributed by metabolic processes into the bloodstream, right? We're adding acid into the bloodstream. And then that dissolves in water to form our carbonic acid, right? And then if we have too much acidity, then that bicarbonate will bind up the extra protons. But then as we move from the middle to the left again, that also represents breathing out, right? Blowing off the carbon dioxide and leaving water behind. So it's a little bit of both. Are y'all tracking with me so far? So this part gets a little bit confusing to some folks because, you know, this represents several different things all at once. It's a little bit abstract. So over here, right, we have the bicarbonate buffer system doing its thing, right? But finally, when that blood makes it to the lungs and we breathe out, we take this and we move over here. And this actually leaves the body, right? And since it's left the body, it doesn't need to be buffered anymore, which means the change that is made to the blood chemistry as carbon dioxide leaves the body is permanent, right? Of course, it's a dynamic system, so we're always adding in more acid somewhere, and we're always breathing out more carbon dioxide from the lungs here. So if we end up with increased carbon dioxide levels in the blood, those medullary chemoreceptors in the medulla oblongata, the brain stem, right, are going to increase respiratory rate and increase respiratory depth. So you're going to breathe faster and deeper, which means you're going to take in more oxygen, but you're also going to release more carbon dioxide, breathe out more carbon dioxide, right? So that's a That's our physiological buffer system, our respiratory buffer system. So I don't really like the word buffer here. I prefer to say respiratory regulation of pH, right? Because the respiratory system is not merely binding the acid and holding it in the body, it's actually removing it, right? So in that sense, it's not truly buffering. So we have a similar reaction. We have other chemoreceptors, peripheral chemoreceptors, in the carotid sinus here in the neck is one location for them that detect pH and not just carbon dioxide levels, right? And so by the same token, if we become too acidic, right, we say we are acidotic, a condition of abnormal acidity, right, acidosis, then we can have the same reaction, right? We can increase our respiration in order to get rid of more carbon dioxide, which is the same thing as removing acid from the blood, right? Which is going to reduce the hydrogen concentration, which is the same thing as reducing acidity or pushing pH upward on the pH scale here. As an aside, your body does not directly measure oxygen concentration in the blood, right? The thing that makes us want to breathe in is the medulla oblongata detecting high concentrations of carbon dioxide, right? So you may be aware that there are people in the world that free dive for pearls and they go real deep in the water and they can hold their breath for 15, 20, 25 minutes, right? It's really cool that human beings can adapt and do that, right? The danger is they've suppressed the reaction to the high concentration of carbon dioxide to hold their breath, but we have no signal telling us when we've run out of oxygen, right? So if a free diver pushes it too far, they just lose consciousness 25 meters under the water. Usually that results in their death, right? You just black out because you don't have enough oxygen to keep your brain working. So hopefully that helps you remember some of that, right? Now, if we have the opposite condition, we have an alkalosis or a condition of abnormal alkalinity in the body, right? The brain will do the opposite. It will slow down our breathing and allow more carbon dioxide to build up, right? And in that way, regulate the pH levels there. So we've always got to talk about homeostasis, right? What happens when the body fails, when it can no longer adapt, right? If we've got something like emphysema or COPD or asthma, right? That represents some sort of malfunction with the respiratory system. So in the case of obstructive pulmonary disorders, right, we can't blow out enough air fast enough. It reduces our ventilation, right? That's hypoventilation. And so it restricts our ability to blow off carbon dioxide, right? And that's going to push the acidity, right? We're going to have more acidity, and that's going to push the pH down. And so then we have to compensate for that in other ways. Or as it says here, we're having a panic attack or an asthma attack. We're hyperventilating, we're breathing very quickly, we're going to lose a lot of carbon dioxide very rapidly, right? And our pH is going to go up. Okay. And so those are examples of conditions that can cause what we call respiratory acidosis. So an abnormal condition of acidity caused by a respiratory malfunction. So respiratory acidosis then is caused by hypoventilation or abnormally low exchange of air into and out of the lungs, right? Because if we cannot exchange air in the lungs, we cannot remove carbon dioxide from the system. And so that carbon dioxide, because dissolved in water is carbonic acid, is going to cause increased acidity. Right? The opposite is true, respiratory alkalosis, an abnormal condition of alkalinity caused by respiratory dysfunction, right? If we're hyperventilating, we're blowing out too much carbon dioxide, losing too much acid too rapidly, and so the blood pH is going to become too high, too alkaline, too basic. Now, we can also have metabolic acidosis, right? So again, an abnormal condition of acidity caused by some metabolic impairment, right? Kidney dysfunction is one of those, right? Renal disease. If your kidneys are not concentrating your urine appropriately, then you need dialysis to remove these metabolic wastes from your body. If you have real bad diarrhea, and I'm not talking about like, you know, 2 instances over the course of a few hours. I'm talking about you got the C. diff, you got the food poisoning, you got the salmonella in a bad way. And it's bad for 2 or 3 days, right? Every couple hours, every time you try to eat something, right? You're going to lose a lot of alkaline substances, right? A lot of your digestive juices, apart from stomach acid, are alkaline. You push a lot of that out, right? And you have to make up for that somewhere. Right? We lose a lot of alkaline substances, a lot of bicarbonate, then we become too acidic. Diabetes can cause a condition called diabetic ketoacidosis. Real starvation can lead to a similar condition. And I'm not talking about fasting for a couple of days, right? And drinking some water. I'm talking about actual starvation, right? Where your body starts to eat your organs for energy. Right? Metabolic alkalosis, right? An abnormal condition of alkalinity caused by some metabolic problem, right? Having your stomach pumped over and over again, or again, you get the salmonella real bad and you're puking for 3 days, right? Keeping a bucket by the bed type situation. You're losing a lot of stomach acid. Well, how do we make stomach acid? Well, we take hydrogen protons, we combine it with chloride, and we make that in cells in the gut. Right, so we got to pull that from somewhere. That means we're removing it from other parts of the body. Certain diuretics, right? These are drugs that make you pee a lot if you're retaining water. A lot of them, particularly the ones that have used in the distant past, would cause a lot of potassium loss. Right, mostly we use potassium-sparing diuretics now. Right, if you take that bottle of milk of magnesia and drink straight from the bottle, or you're eating those tons like they're candy, right? You're eating a lot of alkaline substances. That's going to throw off your balance, right? So those are all conditions there. Need to be able to identify these different conditions, right? Respiratory and metabolic alkalosis and alkalosis, and some of the reasons that they might occur, right? Also, be able to think through how the body is going to compensate. If we have a metabolic acidosis because we have really bad diarrhea, that might increase our respiratory rate, right? In order to reduce that acidity in our bloodstream. Okay. So, just some food for thought. All right, we got to move right on to the next thing. Please share any questions you have right now. Some of the things that might cause metabolic and respiratory alkalosis and acidosis, and then how the body might compensate while you're in the middle of it. Yeah. You could see that equation on a test, right? Generally, it'll be part of the question. So you just need to understand what's going on, right? What it could represent. Typically, the answer to those questions will be explaining what is happening, right? So, yeah. Okay. Oh, that was here. So let's try that again. looking at this schedule again right here. Am I opening up the right thing at all? Yes, I am. Today is the 28th. So yeah, we need to get right along. We need to get through all of lecture 4, and then we're supposed to start lecture 5 today. I usually am able to actually teach you protein synthesis in class. Because we missed that day, we may run out of time and you may have to just watch the pre-recorded lecture video on your own time. All right. That's scheduled for the 9th, right before our first lecture exam. I am not going to be here for this Friday's tutoring. That's going to be Dr. Dorman. But typically, this is the last lecture before the lecture exam. So if you want to go ahead and look at it this week and then bring your questions to Dr. Dorman on Friday during the tutoring time, that would be an option for sure. All right. Golly. All right. So I'm not going to waste any more time. We've got to get to it. All right. Friday from 11 to noon. Room 1082, I think. The one right next door, right here. I think we are attempting to record it. Yes, last week was the first one, and I didn't record it. I was kind of fighting with Teams and the whole automatic camera that's supposed to be following you around and letting people into the Teams lobby. And I think I've got all that figured out now, so we should be able to make it happen.
How can we watch it?
I'll put a link to the Teams meeting on Blackboard for you. I'll post an announcement. So anybody should be able to just log on and join the meeting and view it if you can't be here in person. So, all right. Ooh, organic compounds, right? Organic compounds are compounds that we generally consider to be products of life, although not all of them are. Carbon monoxide and carbon dioxide are carbon-containing compounds that are not considered organic, and things like methane can exist, right, without any proof of life being involved too. But if a molecule has carbon and hydrogen pretty much all the time, it's considered to be an organic compound here. So for our purposes, right, in the human body, the building blocks are carbohydrates, lipids, proteins, and nucleic acids, right? Most of these are polymers of some type. What is a polymer? You may have heard that term used in reference to plastics, and that's absolutely correct. It's just a long repeating chain of smaller pieces, right? So a starch, for example, is a form of carbohydrate made by plants, and it's just a long repeating chain of sugars bound together, right? It's a very large molecule. Plastics are generally the same way. It's the same type of molecule just bound together in repeating chains there. We're going to go back to those chemical reaction types we talked about last week or the week before. Decomposition reactions and synthesis reactions, right? We're going to call this dehydration synthesis because when we plug two things together, we're going to remove a water molecule from the system. And then hydrolysis, right, is that decomposition literally means to cut with water. So we're going to add that water molecule back and split those pieces off again. So up here on the top, right, here's our example. We have two monomers, two individual building blocks, each with an OH group hanging off, right? As they chemically react and bond, we pull a water out there and they remain linked by a covalent bond with oxygen in this case. And then hydrolysis is the same reaction in reverse, right? By flying a water molecule in and cutting them apart, that water molecule will then bond in its original place, right? An OH and an H, allowing those two pieces to be decomposed there, right? So our example here If we take a glucose and a fructose, both 6-carbon sugars, and we bind them together into a disaccharide, right? Dehydration synthesis occurs and we end up with table sugar or sucrose, right? And as you can see, that's an example of a reversible reaction. In our body, right, when we eat sucrose, we have enzymes in our mouth and in our intestines that will break that bond and break it down to glucose and fructose, which we will then absorb into the bloodstream there. So the word carbohydrate means carbon with water, right? Hydrated carbon. And we see C, H, and O, carbon, hydrogen, and oxygen. The generic formula for a carbohydrate is CH2O, right? And that is to say there are twice as many hydrogens as carbon and oxygen exist in equal amounts, right? So C6H12O6 is the generic formula for any 6-carbon carbohydrate, right? C5H10O5, right? It's still a carbohydrate, just a 5-carbon sugar. So monosaccharides, these are the monomers, right? This is the smallest thing that is a carbohydrate. A disaccharide, like sucrose, is 2 of those monosaccharides plugged in together. And then anything more than 2, we call a polysaccharide here. So our monosaccharides that we'll be dealing with here, we have hexose sugars. These are all isomers of each other. The chemical formula is the same, C6H12O6. You can take the time to count them up if you wish. Glucose, fructose, and galactose, right? And then we have our pentose sugars, 5-carbon sugars, ribose and deoxyribose. Those are going to show up again later. What are carbohydrates used for? Essentially, they're jet fuel, right? They're easily available energy. We can store them as glycogen in the liver, right? We can put them through cellular respiration, that is glycolysis and the citric acid cycle and the electron transport chain to create ATP. And we can even convert them into fat, right? If energy is in excess, right? So those disaccharides like sucrose, they are too large to be passed through a cell membrane and absorbed into those intestinal cells, right? Those simple columnar epithelial cells with microvilli. And so along the brush border of those cells, we have what are called brush border enzymes, things like sucrase and lactase, and right, that are going to break apart things like lactose and sucrose into their simple sugars. And so they can be absorbed. Human beings are pretty much the only species that I'm aware of that continues to drink milk and dairy products after infancy, right? Some of us are blessed and maintained our lactase enzymes into adulthood. A lot of you probably did not, right? If you are lactose intolerant, that is simply because you no longer make lactase, which is the enzyme that breaks it apart. And so you have a disaccharide in your belly, and because you can't do anything with it, the bacteria and yeasts inside you do something with it, and they make a lot of gas, and that's what kind of makes it painful and bloaty, right? So, there we go. So here are chemical examples of sucrose, maltose, and lactose here, right? Again, these are isomers, right? The chemical formula is the same, right? It's a disaccharide, 2 6-carbon sugars plugged together. So the chemical formula would be C12H24O12, right? But they're arranged slightly differently, right? That's what an isomer is. Same atoms, just in a different pattern. And even though it's all the same atoms in just a slightly different pattern, right? It requires completely different mechanisms to break them down, completely different enzymes. Now, a polysaccharide, we have glycogen pictured here, right? It's how animals store carbohydrate. We store it in our muscles and our livers primarily, and we take glucose and we plug those glucose molecules in long chains, right? That's glycogen. Starch is how plants store sugar for energy, right? And that's why you find it in the kernels of corn, so on and so forth, and all the grains. Cellulose would be the structural sugar that plants use to form things like cell walls. Now, lipids. These are fats, right? They also contain carbon, hydrogen, and oxygen, sometimes some phosphorus. We have a lot of different kinds. Triglycerides are the type of fats we typically find in the blood. Phospholipids are mostly a triglyceride, but instead of a third fatty acid, they have a phosphate group attached. Steroids, right, are all derivatives of cholesterol, which is a fat, and then eicosanoids are tiny little fats that primarily function in cell signaling. What does fat do? Right? Energy storage is a big one. It's very energy dense, a lot of calories per gram, bless you. It can help insulate our body, right? Brown fat is metabolically active and actually produces heat and then provides that physical cushion. Behind our eyeballs, for instance, we have a little fat pad helping to cushion and insulate the eyeballs here. So this is a triglyceride. Right over on the left-hand side, we can see the glycerol backbone made up of a 3-carbon chain. And then we can attach fatty acids to this glycerol, right, by removing water. So again, dehydration synthesis. At each one of those oxygens on glycerol, right, we remove a hydrogen and we remove an OH group from the end of our fatty acid. Water left over, and that allows those bonds to be formed. Again, dehydration synthesis. Now, when we're looking at fatty acids, a saturated fatty acid has a hydrogen atom bound at every available site on every available carbon in the chain. So while we're looking at triglyceride here, if we just take a single fatty acid tail, that little zigzag line, at every point on that zigzag line, that represents a carbon atom with 2 hydrogens bound to it. Its other 2 bonds are taken up by the carbon on either side. Carbon can make 4 covalent bonds, right? So all 4 are bound up. In a saturated fat, these tend to be solid at room temperature. So if we're thinking about things like butter and lard, or maybe even coconut oil, right? These have different mixtures of saturated and unsaturated fatty acids. The higher percentage of saturated fatty acids, the higher the melting temperature, the more likely it is to be solid at room temperature, right? Oils, on the other hand, right, are unsaturated, or they are high in unsaturated fatty acids, right? Are heart healthy oils, quote unquote, like olive oil or avocado oil, perhaps, things that are naturally high in fat. If you wanted to call things like canola oil and vegetable oil and rapeseed oil heart healthy, I might have to challenge you to a duel. But the point here is that this fatty acid, instead of being bound to 2 hydrogens and 2 carbons, there is an open spot. One of those hydrogen atoms does not exist, leaving 1 bond available for a double bond between 2 carbons. That double bond puts a little kink in the chain, right? Causes it to deflect off to the side, right? And because it has this little irregularity, it doesn't fit neatly in with all of the other pieces. If you want to think about it like crystals, right? You start putting random stuff in a crystal structure, the crystal structure doesn't form, right? So it's less likely to be solid at room temperature. This is a phospholipid. If you look very carefully, you'll see that it is made of the same glycerol backbone, 2 fatty acid tails, but where that 3rd fatty acid might be attached, we have a phosphate group. That is a phosphorus bound to 4 oxygen atoms, And then we have a little nitrogen containing group out on the top. Now, phospholipids are important because that phosphate head likes to be in watery environments. It is hydrophilic. The fatty acid tails, being fats, right, they're hydrophobic. And we find these compounds in our cell membranes, right? The membranes of the cells themselves and of many of the organelles within the cells are made up of a double layer of these phospholipids in the arrangement that you see over on the right, with the hydrophilic heads pointing to the outer surface and the inner surface, right? Meaning they're compatible with the watery environment inside the cell and the watery environment outside the cell. And then those hydrophobic tails being repelled by the water are oriented toward the inside of the membrane. Steroid hormones, right? Steroids are all some derivative of the cholesterol here, right? We can see we've got some hydrocarbon rings, and then you see that familiar zigzag up on top, right? That represents a small little fatty acid tail, right? So this is the generic structure for steroid hormones and cholesterol here. Cholesterol is embedded within your cell membranes, and it allows for the cell membrane to be flexible and not too rigid. It's part of vitamin D synthesis. Vitamin D is sort of a misnomer. Vitamin D is really more of a hormone in the body. It acts more like a hormone than a vitamin. Um, yeah. Yeah. And so fat soluble vitamins A, D, E, and K, because they're fat soluble, you don't pee them out, right? They don't get dissolved in water and excreted in urine. And so they can build up to toxic concentrations more easily. Whereas things like vitamin B, any of them, and then vitamin C, right? You can basically take vitamin C until it gives you the runs, and you're not going to suffer any ill effects other than, some watery feces, right? It's not going to hurt you. You can just get rid of it. If you take too much vitamin B, you're just making some expensive bright yellow urine. But there is that. All right, now proteins, right? In plants and animals, proteins make up much of the structure of the body. They are the most varied of all of these macromolecules, of any of these organic compounds. Every enzyme is a protein. We have tiny proteins for cell signaling. Antibodies are proteins, right? The keratin in your skin, hair, and nails is a protein. The collagen in your skin is a protein. The elastin, the reticulin, so on and so forth, right? These are proteins. Hemoglobin and myoglobin that carry oxygen are proteins, so on and so forth. Thousands and thousands and thousands and thousands of different proteins are created by the body. Notice they're made up primarily of the same three elements: carbon, hydrogen, and oxygen, sometimes a little phosphorus, but what we're adding here is nitrogen. So if you know anything about gardening or growing grass, right, fertilizer, if it's going to green up the grass and make it grow aggressively, is going to have nitrogen in it. Why is nitrogen important? Right, because it's a necessary component of those structural parts of the organism, right? Proteins, like carbohydrates and fats, are polymers. The monomer is the amino acid, and we have 20 different types. There's a specialized bond between these amino acids called peptide bonds, right? A group of 2 amino acids bound together is a dipeptide. 3 is a tripeptide. And then from there, we get into polypeptides, or we just call them larger proteins at that point. We've already talked about how these different groups on the amino acids can act as either a weak base or a weak acid and form part of that protein buffer system inside the blood. And so if we look at these here, right, we have our monomers, the individual amino acids on the left. If we plug them together, we pull out a water molecule, and dehydration synthesis and form a carbon-nitrogen peptide bond, all right, binding those amino acids together. If we are digesting protein that we've consumed, right, we can use our stomach acid and other enzymes to break the peptide bond, adding water, right? And then we can then absorb those amino acids and use them to create our own proteins and use them as building blocks here. Now, proteins, they are somewhat complex. There are whole computer networks dedicated to calculating how proteins fold based on the complex interaction between all the different atoms and all the different amino acids and so on and so forth. So the primary structure of a protein, most of what we're going to consider, is just the sequence of amino acids, right? If we have methionine, alanine, threonine, blah, 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 one after the other, right? That's the primary structure. Now, as that chain of amino acids is leaving the ribosome and it begins to fold up into a two-dimensional or three-dimensional structure, right? Either the alpha helix or a beta pleated sheet, we consider that to be a secondary structure, right? The pattern continues. As these secondary structures begin to interact with each other and form more complex three-dimensional shapes, that's a tertiary structure, right? And then the completed polypeptide, as it interacts with other proteins to form a larger, more complex molecule, that is said to be quaternary structure. So primary structure, right? This is just the chain of amino acids. It says nothing about how it's going to fold and what the eventual shape of the protein is going to be. It's just a list of amino acids here. Our secondary structures are represented here, right? Our options are the alpha helix or the beta pleated sheet. So one of them looks like a spring and the other one looks like the bellows of an accordion, if you will, or the pleats on a kilt. And then the tertiary structure is a more complex organization of those. So the one pictured here is called transthyretin. It's a transporter protein binding thyroid hormone. And then the quaternary structure of this functional transthyretin molecule is made of those four of those tertiary structures bound together, right? This would be the complete protein here. Hemoglobin is another example that's made up of, I think, 4 different tertiary structures. So we've already talked about some proteins in lab, right? Primarily the fibrous proteins, right? Collagen, elastin, reticulin, right? There are also globular proteins. Those tend to be functional, things like enzymes, transporter proteins in the blood, right? And here are some different examples. The structural proteins you're more familiar with already, right? Hemoglobin isn't another example, right? We can see the 4 subunits there binding to oxygen. Our enzymes, those catalysts, are really, really important for their three-dimensional shapes. Contractile proteins in the muscles. Actin and myosin, we'll talk about that not too long from now. And then hormones, antibodies, right? A lot of these things have protein components, right? Neurotransmitters are all proteins, so on and so forth. Okay, so now we've already talked about how pH can cause things to malfunction, right? And that has to do primarily with the process we call denaturation of proteins. You can think of denaturation as a meddling with the natural shape that a protein follows, right? So if pH gets too high or low, that's going to change the environment. That's going to change how those positive and negative charges interact with each other, break hydrogen bonds, and cause those three-dimensional structures to unfold essentially or break apart. And again, because form and function are inextricably linked, right? If you unfold a protein, it stops doing its job, right? So, a lot of times, a partial denaturation, a slight tweaking of the shape of an enzyme because of abnormal pH, for instance, will be reversible. When the pH is restored to normal, the enzyme will resume its natural shape and resume its functional efficiency. But sometimes if it goes too far, it's irreversible, right? If we cook an egg and change the albumin in the egg white from that clear to the edible white, right? We're not going to uncook an egg, right? It's functionally irreversible. We could go into the lab and do some fancy chemistry, but you're not going to eat it afterwards, right? That would be an example. Ceviche is another example, right? We take all that raw seafood, drench it in all that lemon and lime juice and other goodies, and the acidity denatures the proteins in that seafood and changes it from that translucent color to that more opaque white. That's what's happening. We're unfolding, we're essentially cooking those proteins with acid. Now, I've talked to you all about enzymes and catalysts already. This is a good graphical representation of what it's doing, right? A certain chemical reaction needs its reactants to be present in such a way that they can actually interact with each other, but they have to interact with sufficient energy to force the chemical reaction to occur, right? So a catalyst, by binding to reactants in a specific way, will sort of force those reactants together in a way that drastically reduces the necessary energy for the reaction to proceed, right? So it's going to drastically increase the speed of that reaction here. And so this is activation energy. In both cases, we end up with a product, right? But with an enzyme, right? If this is a little round beaker on top of a Bunsen burner, right? We have to apply a lot less heat over here to make the reaction occur. Yes. We have some vocabulary here. Right. We have some enzymes that function entirely on their own. Some enzymes require something called a cofactor, which would be something like zinc or magnesium, right? Elemental zinc or elemental magnesium present to assist it in its job. The coenzyme or cofactor is not typically a part of the chemical reaction. It's just there to interact with the enzyme there. So typically, I can't think of any time when it's not. If it's an organic molecule, then we would call it a coenzyme. So to be honest with you, it's been 20 years since biochemistry and organic chemistry for me. So some of those details have floated away on the wind. And you just kind of need to understand that they exist and what they do. We're not going to get too deep into the details here. What is very important is that enzymes, again, they're very chemically specific, right? If we go back to sugar digestion, right, even though the chemical formula for lactose and sucrose are identical, right, C12H24O12, because those atoms are arranged slightly differently, we have to have a completely different enzyme in order to break them apart into the side, right? It's lactase for lactose and sucrase for sucrose, right? And sucrase will not act upon lactose, right? That three-dimensional structure binding the specific atoms in that specific orientation is very particular, very precise, right? So an enzyme designed to break peptide bonds will not also break fatty acids apart from a glycerol backbone, for instance, right? An enzyme is going to do one thing, it's going to do it very well, and then it's going to move on to the next time it does that thing, right? So, yes. Right. So the first two bullet points here, I've already said to you several different times. This last bullet point, I think, is better visualized with this next slide. So in a simple chemical reaction, where we have two reactants, right? Two substrates, we'll call them, right? They're going to bind to what we call the active sites. This is the site on that enzyme protein that is designed to bond to the substrate, right? As the substrate bonds, right, we've essentially created a new molecule, right? The enzyme substrate complex with a whole new set of electromagnetic interactions between all the atoms. That's going to cause the whole structure to change shape, right? To change conformation. That conformational change is designed to cause the chemical reaction to occur. And then once that occurs, we have another shape change, which causes the end products to be released from those active binding sites. And then once it's released, that enzyme goes back to its resting state with open active sites ready to receive two more substrates. So in this way, an enzyme causes the reaction to happen much more rapidly with much lower activation energy, but it is not itself consumed, right? It's sort of just grabbing and plugging together and letting go right over and over and over again in this example of the synthesis reaction, right? Now the opposite would be true for a decomposition reaction, right? It takes a more complex molecule, grabs it in two places, and then it changes shape and rips it apart, right? So it can go either way, right? But no matter what an enzyme is doing, that's the process, right? So, right. So that's proteins. Nucleic acids. What do we see here? Carbon, oxygen, hydrogen, nitrogen, and phosphorus. So the same 5 elements that we found in proteins. Right? Nucleic acids are, of course, the structural components of our genetic material, our DNA and our RNA, right? DNA stands for deoxyribonucleic acid, right? So we have a nucleotide with a 5-carbon sugar backbone and then a nitrogenous base here, right? And then there's a phosphate group plugged in to the end. So in DNA, we have 4 bases, right? The purines and the pyrimidines. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. Now, a DNA strand is a double helix, meaning there are 2 strands, right, of nucleotides. They're oriented opposite to each other, right? So one would move from left to right, the other from right to left, but they're complementary to each other. They're not identical, right? But each strand is going to complement. We accomplish that by using what we call the base pair rule, right? Our adenine is always going to pair with thymine, and guanine is always paired with cytosine. So if we have a particular chain of nucleotides across the top strand, right, the complementary pair of those strands would be running along the bottom strand. And we can decode either strand here. Now, we have a 5-carbon sugar. In DNA, it's deoxyribose, right? And then, of course, we have our nitrogen bases and a phosphate group here. The primary job of DNA is to provide codes for protein synthesis. So if we need to make hemoglobin, in those cells that produce hemoglobin, right, the machinery in the nucleus is going to unzip that part of the DNA, separate the 2 strands, where hemoglobin is coded for. It's going to read the sequence of DNA nucleotides, right, turn it into a thing called mRNA, right, and there's some little bit of editing that happens. Then that mRNA leaves the nucleus, and we translate that mRNA into a protein, right? We read that code, right, and it determines the order of amino acids that get plugged in together to make hemoglobin. Our structure here, right? The backbone, right? That deoxyribose phosphate backbone, and then the nitrogen bases are bonded to each other. And so we can see here that the two helices of DNA are bound together by the bases with hydrogen bonds in between them. If you look very closely at this diagram, you can say adenine and thymine share 2 hydrogen bonds, where cytosine and guanine share 3. Right? So that's one of the mechanisms to make sure that they are always paired and they always link together in that way. Right? Now, RNA is ribonucleic acid. So the 5-carbon sugar is just ribose, right? You probably already figured that out. RNA is single-stranded. Most of the RNA we're going to find outside of the nucleus in the form of mRNA and tRNA, right? There's some ribosomal RNA that makes up a portion of the ribosome itself, which is the protein enzyme RNA complex that is the manufacturer. It's the little machine that punches the little amino acids together, right? And so RNA, if DNA would be, something locked in a vault, the RNA would be the message that gets sent out, right? The DNA is too valuable to leave the protection of the nucleus, right? So we make a little transcription, a little copy of it, and send it out into the world, right? We don't pull the Declaration of Independence out of its glass case in the museum, right? We have reproduction prints of it, right? The RNA would be like a reproduction print. Now, of course, I immediately thought of that Nicolas Cage movie. Now, ATP, adenosine triphosphate, is the energy currency of our cells here. It is immediately usable. We break a phosphate bond, and then that energy can be directly used to activate enzymes, open gates, power pumps, cause muscular contraction, so on and so forth. Now we have an adenine nitrogen base, again, a ribose sugar, so it looks a lot like our nucleic acid, right? But we have 3 phosphate groups bound here. Now the bonds in between the phosphate groups are called phosphate bonds. Very clever, I know. But they store an immense amount of energy. So ATP is the preferred currency of the cell. But if we run out of ATP, we've broken a phosphate off and it's all converted to ADP, or adenosine diphosphate, we can break that second phosphate group off and leave ourselves with adenosine monophosphate. And through the electron transport chain and other mechanisms, we can add phosphate groups back onto that, right, and regenerate ATP, right? But it is metabolically expensive, right? All of the glucose we burn through pretty much serves the function of creating that ATP. Now it functions through what we call phosphorylation. So we're breaking the phosphate bond. That phosphate group is going to bind to something else and transfer that energy here. So for example, right, we have a pump in our cell membranes called the sodium potassium pump, and it is constantly working to pull sodium out of our cell and bring potassium back in. But it does it against the concentration gradient, meaning there's a lot of sodium outside the cell, so it has to push pretty hard to push more sodium outside of the cell. So it's not just something that's going to happen constantly. We actually have to spend energy, right? It is a proper molecular pump, right? And then we have other examples of opening gates and channels. Sometimes we'll use ATP and similar molecules to transfer messages from outside the cell membrane inside the cell membrane and then activate what we call second messengers that are going to cause other things going on. And we'll talk about some of those processes later in the semester. Woo! Still going. Inorganic phosphate. Yep. That's the phosphate group that got broken away from ATP and released as energy. All right, what are we doing for time? 6:16. All right, I'm not going to do anything meaningful in 4 minutes. So I'll see you at 6:30 over in the lab. And we'll just try to do all the cell structure on the second, if we can get through it.